So today we will look at a new topic, which is the importance. We are going to talk about the importance of buffers in a biological system, because we are talking about analytes, we are talking about proteins, we are talking about, say, cell organelles. How to we studied cell fractionation, centrifugation. In that, we are all discussing about how to separate a particular organelle, or how to separate a protein or any other biomolecule from a mixture of other compounds, okay, from the mixture of another other molecules. So in all of that, buffer system is extremely important. Now buffers, because when you're talking about a cell and a cell organelle, the cell organelles are inside the cell, inside in the cytoplasm, and then that cytoplasm is constituted of, say, majority component of a major component of cytoplasm is water. Or most of the living creatures, the most important constituent of these creatures is about water, and that is 70% of the mass. Okay, so that's quite a bit of water. Now, is it only water? No, this water will have some ions. This water will have some important salts. Okay, so this cytoplasm, or the water-based cytoplasm, or you can say the aqueous system inside the cell, is so important because all the biological reactions that are happening in the cell is called as a factory, because there are, at the same time, there are so many reactions in parallel going on. So you need a medium for all these biological reactions to happen. So these biological reactions happen in the aqueous medium. Okay, then all the aspects of the cell structure and function are adapted to the physical and the chemical properties of this aqueous system, which has largely water. So the particular structure of a cell organelle, maybe it is a mitochondria, ribosome, or endoplasmic reticulum, nucleus, or we're talking about, say, a protein, we're talking about any other biomolecule, we're talking about drug intermediate is there, or metabolite is there, which is being produced by the cell, or it's a hormone. All the aspects of the structure and the function of that particular analyte is dependent on what is the composition, the physical and the chemical properties of the water or the aqueous system, which is there in the cell. Okay, now since we're talking about water, water is H2O, and you know that it splits up as H plus and OH minus. Now this H plus and OH minus concentration influences the structure, the assembly, and the properties of all the macromolecules in the cell. And also, why is water chosen, or why is water naturally forms this aqueous base? Is because water is able to dissolve most of the charged molecules. It's a polar solvent. It can actually dissolve abundant charged molecules. So that's why, and we all know that it is also called as, say, a universal solvent kind of. Yeah, it has this name. So it can dissolve many charged molecules, and that's why water-based systems are you encounter water-based systems in all the living creatures. Okay, several biological molecules such as proteins, certain vitamins, steroids, and phospholipids, they contain both polar and non-polar regions. So there are certain regions which, in lipids, proteins, vitamins, steroids, or in all these biomolecules, in all these macromolecules, which are called as hydrophilic and hydrophobic. So a certain part of these proteins has affinity towards water, so they are known as hydrophobic. Then the other end might be hydrophilic. Okay, so sorry, I think I said the opposite. Hydrophilic is water loving, hydrophobic is water hating, or something, you can say. That the molecule is drawn towards water, then you say it's a hydrophilic molecule. If the molecule repels water, if the molecule goes away from the water molecule, then this is hydrophobic. Okay, so since they contain both the components, philic and phobic components, they are also called as amphipathic molecules. Okay, Now, before we move on to the buffer system, because in at every stage in a biochemical experiment, or in at every stage where you're extracting a protein, where you are centrifuging, where you are actually subjecting the protein to column chromatography, then electrophoresis, everywhere you're going to see, and then further you're going to do, say, an enzymatic assay to check the activity and the stability. At every step you're going to need a buffer. Now this buffer is very specific for proteins. uh, every protein has a specific requirement for buffer and temperature. We are going to see that also, that how the change in temperature and how the pH change affects the structure and the function of the particular analyte of a biomolecule of a protein. Okay, all enzymes are protein, but all proteins are not enzymes. So this is one of the statements that you must remember. Now, before we go on that, we'll just get acquainted with what is dissociation constants of weak acids and bases. You have studied what are acids and what are bases in your school syllabus. The strong acids and the strong bases, when they are added to water, they dissociate completely. Okay, they ionize completely. So then we call them as strong acids and strong bases. Something like sulfuric acid, hydrochloric acid, as soon as you add them to water, they can ionize. They ionize as H plus Cl minus. Okay, like this. If you add NaOH, which is a base, so Na plus OH minus, K plus OH minus. So that these are strong acids and strong bases. So that's why they dissociate completely. But when you add a weak acid and a weak base, or a weak base, so the weak acid, let us consider an example of acetic acid. Acetic acid is also something that we can consume. These are also consumable acids, the weak acids. Okay, so they don't completely ionize. So that's why, or they don't completely dissociate in a solution. So biological systems encounters only weak acids and bases. In our biological system, in a cell, when a particular reaction is happening, there are no strong acids that our biological system has. The system only has weak acids and weak bases. So weak acids and bases do not completely dissociate in a solution. They exist. Okay, they do exist because they are required. Otherwise, there is no pH maintaining happening if there are no acids and no bases in our biological system. And if there is no pH, then there is no activity possible, the enzyme activity, and hence there will be no reaction for the reaction. So they exist, they do exist. These weak acids and weak bases, they do exist instead as an equilibrium mixture of undissociated and dissociated species. So some of them will dissociate and some of them will not dissociate. So if you're talking about acetic acid, this is CH3COOH. So you can see this is K1, this is K2. So it's a reversible thing. It's a reversible reaction that acetic acid will dissociate as H plus and CH3COO minus. Okay, and it might also revert and form an acetic acid molecule, acetate molecule also. So here you will see that this is CH3COOH has split into hydrogen and acetate. Okay, further hydrogen and acetate can go back to form acetic acid also. So this K1 and K2 are rate constant. Okay, rate constant in how much time they dissociate, in how much time they get back, they associate. Dissociation, association. So K1 is for dissociation, K2 is for association. They come back. So these in an aqueous solution, the acetic acid is an equilibrium mixture of containing both these ions in equal quantity, and along with it, it will also have undissociated acetic acid. The equilibrium between these species can be expressed as this equation here. As I told you, K1 and K2 are representing the rate constant for association of acetic acid. Okay, and you can see now that the rate of dissociation of acetic acid is, because we're talking about rate, then the factor of time comes into picture. That's why it is dt here. So acetic acid by time is dependent on the rate constant of dissociation. So the rate of dissociation of acetic acid is dependent on the rate constant, that is K1, and the concentration of acetic acid depends on the rate constant. So the splitting depends on K1, the formation of acetic acid depends on K2. So if you look at this, you have included, or you're multiplying this with K1. K1 is nothing but the dissociation constant. Similarly, the rate of association. So how quickly are these two ions, H plus and CH3CO minus, how quickly are they getting together and forming a acetic acid molecule? So here we are seeing that how quickly does it dissociate to give you H plus and CH3COO minus. Here we are seeing that in what time these two ions come together and they form acetic acid here. Okay, so here the rate of association, that is the rate of formation of acetic acid, is dependent on the rate of association. The association constant is K2. Okay, so more the ions of H plus and CH3COO minus, more will be the formation of acetic acid, plus time is a factor here. Okay, so concentration of acetate and hydrogen ions, that can be expressed. So when you mention the concentration, you always mention them in square brackets like this. So right now we are talking that two different ions are formed. One is H plus and CH3COO minus. So we are indicating, or we are showing them in two separate brackets, because these are two, one of them is positively charged, one of them is negatively charged, so you have to show them separately. So this is this one whole acetic acid which splits into H plus and CH3COO minus. So rate of dissociation is how fast will be the splitting into H plus and CH3COO minus. Rate of association is how fast is going to be the bonding and forming of acetic acid. Okay, now since the rates of dissociation and reassociation, because it's an equilibrium condition, we are talking about weak acids. So either when you add it to an aqueous solution, the acetic acid will be in an equilibrium mixture of H plus and acetate ion and some undissociated acetic acid. So you will see that you can write it in this way, K1 CH3COOH, because here this is for the dissociation. Okay, this is the dissociation constant. And K2 is the association constant. Okay, so you can write this equation in this way. Then if you take K1 by K2, bring the constants on the same side, and you can then rearrange the equation. So it becomes like this. And hence now this K1 by K2, that is the dissociation constant by association constant, gives you a new constant which is known as Ka, and this is known as the equilibrium constant. Okay, so the equilibrium constant of, say, acetic acid is written like this. And if you know the concentrations in numbers, then you can also calculate what is the equilibrium constant of this particular acid. Okay, or if it is a base, then you can calculate that. This equilibrium constant now can be rearranged as, so you can very well rearrange it like this also. So in the previous equation you saw it was Ka is equal to H plus CH3COO minus divided by CH3COOH, right? So what did you do? You kept the H plus, you took the H plus on one side, and now you have rearranged the equation and it becomes this. So the hydrogen ion concentration now can be expressed, it can be calculated in the terms of equilibrium constant. So if you know the equilibrium constant, if you know these concentrations, then you can easily find out what is the concentration of the H plus ion. So the equilibrium constant for ionization reactions is called as ionization constant or dissociation constant. So with this, we can move on to what is like, what is a buffer system? We just saw it's an aqueous system, okay? But what is the use of this aqueous system? The aqueous system is important because it resists any change in the pH. So this pH factor is so important for the biomolecules, for a proper functioning of a cell, for proper retaining of the structures of the biomolecules. This pH is extremely important because it resists the changes. This buffer is so important because this buffer system is going to resist any change in the pH. So suppose when small amounts of acid or bases get added to this buffer system, then the change is not going to happen there. Because if the change happens on addition of small amounts of acid and bases, then if the pH changes, then the biomolecule structure will change, the biomolecule's function will change. It may not catalyze that particular reaction. And if that reaction does not happen, then the cell will not get that particular product, which is important for the functioning. So a buffer is an aqueous system which resists changes in the pH when small amounts of acid bases are added. Buffer solutions are composed of a weak acid. So there is always weak acid which is acting like a proton donor, and its conjugate, which is a proton acceptor. So there is a donor and there is an acceptor. So the buffer will always have a donor and an acceptor. So you will have a weak acid and its conjugate base. So that base is going to be the proton acceptor. Buffering results from two reversible reaction equilibria in a solution wherein the concentration of proton donor and its conjugate proton acceptor are equal. We just saw what happens to acetic acid. So if you're talking about the dissociation, so acetic acid is also used as one of the components in the buffer system because it is a weak acid. Okay, so it will have a conjugate base. It will have a base, and there is like sodium acetate buffer, for example. Okay, sodium acetate buffer in which now acetic acid and the other component, other base will be NaOH, conjugate base. So when you mix both of them in a certain proportion, you are going to get a buffer. Okay, and now here in this case you saw it will dissociate. It will give you CH3COO minus, it will give you H plus. Okay, so here you will see proton. Yes, so this plus and this minus. So this dissociation this is going to happen for NaOH also. If you're going to mix both of them to make a buffer, so Na plus and OH minus. So then this is a two reversible reaction. Now when they split, when acetic acid splits, when NaOH splits in the aqueous solution, they're going to be in equilibrium. Okay, and the concentration of the proton donor and the concentration of the proton acceptor are going to be equal in a buffering system. So in a buffer system, when the concentration of acetic acid and acetate ions are equal, addition of small amounts of acid or base does not have any detectable influence on the pH. That is what the buffer system is used for. So if you have a buffer and small amounts of acid or base is added to that buffer system, then you will not see any detectable influence on the pH. That is because this capacity of the buffer to maintain the pH is going to affect or is going to maintain the rate of reactions also of the enzymatic reactions in the cell. So if a cell is always at pH 7, if a cell is at pH 7, and this is the optimum pH for the reactions, optimum pH for biochemical reactions. Okay, so if this is the buffering system, if this is the buffer which maintains the pH at pH 7, and as a result of some biochemical reactions, if there is some, say, a byproduct which is formed, say it's an acid or base, so where will this go? It will remain in the cell, no? Whatever acid or base as a byproduct is formed as a result of this reaction, of this biochemical reaction. So if these components are formed, where are they going to go? They will be released in the cell. Later on, the cell will dispose them, but for the time being it is going to be in the cell. So if they keep on adding inside the cell, it means that the pH will get disturbed. Why? Because addition of acid is happening, or as addition of base is happening. But because of this buffering system within the cell, the detectable influence on the pH, you will not be able to see that. Why? Because this buffer is resisting the change in the pH. Whatever acid and base is being formed as an outcome of any other reaction will get added to the cell, but because of a good buffering system, you will not see any change in the pH. Okay, so it's an extensive property. It offers protection against the changes in pH. Now, buffers with higher concentration offer higher buffering capacity, which is quite obvious. pH is dependent on the absolute concentrations of buffer components, but their ratio. Okay, so their ratio is important, and it is pH is always dependent on the accurate concentrations of the buffer components. So if you're making a sodium acetate buffer, so what is the concentration of acetic acid, what is the concentration of NaOH, that is very, very important, as well as their ratio also is very, very important. Okay, now we'll talk about biological buffers. So biological buffers are the buffers which are there in, like, as I said, that most of the biological systems are made up of water, and these water will have some ions, it will have some salts, it will have a very nice buffering capacity, which will be very, very helpful for the biochemical reactions to happen. So the pKa should reside between 6 to 8. Okay, they should exhibit high water solubility and minimal solubility to the organic system. So the biological buffers that you're going to use, you can also artificially make these buffers. Okay, so if you need pH 7, the very commonly used buffer for in a lab which is working on protein, and if suppose the protein or the enzyme that they're working on is stable at pH 7, because they're recreating the conditions from where this protein has been isolated. For suppose this cell, bacterial cell, from which you have isolated a protein in the lab, the protein was, say, inside here, you grew the cells, and now you have this protein isolated. Okay, but here inside the cell the pH was 7. So when you're doing all the purification steps, you're working in the lab with this particular protein, then you will also have to maintain the pH as 7. So in your test tube, in your column, in the electrophoresis, so gel electrophoresis you may not, because there you're only analyzing the purity. But if you're doing a preparative gel electrophoresis, if you're doing a column chromatography, there you will have to maintain the pH. Okay, so biological buffer, you must know what is the pH of your source from where you are extracting, and then you can recreate those conditions back in your lab. Okay, they should exhibit higher water solubility. So yes, so here the water solubility of your buffer system must be very, very high. Whatever components are, whatever, if you want to add something more in your buffer system, okay, if you want to add certain component in your buffer system, it should be easily soluble, but it should not have solubility, or it should have minimal solubility for organic solvents, because biological systems do not work in organic solvents mostly. So there are certain enzymes like, say, lipases. They are known to work in organic solvents, but most of the enzymes, they are active at in the aqueous phase itself in a proper buffer system. Okay, so the pKa is a number. Actually, it's a number that describes the acidity of a particular molecule. So if you are making acetic acid buffer like sodium acetate, so acetic acid will also have a certain pKa. If you're making another acid, tris HCl, HCl is also an acidic component there, so that will also have its own acidity of a number. So that is a pKa number. So the pKa number should always reside between 6 to 8 for a biological buffer. That is a requirement. Now that buffer the biological buffer should not permeate the cell membranes, because if it permeates, it will go out. We know that the cell membrane is semi-permeable, so the buffer must stay inside because that is its job, that is its role, because it is providing a particular pH for the reactions to happen. So what will happen if the buffer leaves the cell and goes out? Obviously the cell will be without any buffering system, so proper reactions and will not happen. So it should not leave the cell, it should not permeate the cell membrane, it should not exhibit any toxicity towards the cells, it should be a non-toxic, it should be an inert component. The buffer should be no, it should not exhibit any toxicity to the cell itself. No harm should be done to the cell by the buffer. It should also not interfere with any biological process. So it has to be inert, that it should not interfere in any biological process. The salt effect should be minimal. However, salts can be added as required. So as I said, if you want to add some more salts to the buffer, you should be able to do that because it's a water-based system and the solubility of this system is high. So salt will dissolve. Ionic composition of the medium and temperature should have minimal effect of buffering capacity. So change in temperature or change in the ion concentration, it should not have any effect on the buffering capacity. So the whole idea or whole purpose of buffer is to resist any change. So if you try, or if you, is it is because of certain conditions in your experiment, if at all the ionic composition or the temperature is going to be uh, is going to come into picture, if these are the two parameters which are unavoidable, okay, you have to have them, otherwise you cannot proceed with the experiment further, then these factors should not affect your buffering capacity. Buffer should be stable and resistant to enzyme degradation. Okay, so these buffers should not be responsible for enzymatic degradation. Buffer should not absorb either the visible or in the UV range. This is again very, very important because, as I said, spectrophotometric analysis is very important when it comes to biomolecules, because you are trying to do an assay, you're trying to find out the activity of the cell, of the protein. You'll have to do an enzymatic assay. Okay, then if you do an enzymatic assay, you are going to read it in a spectrophotometer. Since you are working with enzymes, all of these reactions are going to be in a buffer system. So buffer is going to be a part, but your analyte or your biomolecule is going to be suspended in the buffer, and then you're going to keep that entire thing in the spectrophotometer for reading the absorbance. So here the buffer should not be absorbing any of the visible or the UV range rays. Okay, that's because if that happens, then you are not getting a true reading. You're not getting a true reading. That's why what we do is we usually have two cuvettes. So here, suppose one of the cuvette is going to be your sample and one of the cuvette is going to be your control. So in the sample, it is always the buffer plus analyte, because your analyte will be unstable if there is no buffer. Analyte will be maintaining the conformation, maintaining the active state only because of the buffer. So it must be suspended in the buffer, and the control will be only buffer. Okay, so you take the reading of 2, and whatever reading you get, it is actually the whatever noise or whatever absorbance. So buffer ideally should not absorb anything, but in this case we are just making sure that we are nullifying, or we are we are going to subtract the absorbance that is going to happen by the buffer. So whatever reading you get is the reading which is minus any interference of the buffer. Okay, so buffers used in cell cultures, isolation of cells, enzyme assays, and other biological applications must possess their distinctive characteristics. So for different pHs, different buffers are used. Different buffers are used for different purposes. So if you're working with cell culture, there will be a different buffer. If you're doing isolation of cells, you're using a different buffer, because there are those components in the buffers which will be responsible for, say, maintaining the structure of a particular component or particular cell or enzyme or other biological applications. So the components are having distinctive characteristics, and that is why you have specific buffers for specific purposes. Okay, they should low interference. That is one of the important characteristic of the buffer. Okay. Now, effect of temperature on pH. We are talking about buffers because buffers are related to maintenance of pH. As I told you, while you're doing certain experiments, sometimes it gets unavoidable that you might have to incorporate heat, or you might have to heat your system, whatever you're working on. Or probably if you are sonicating, last time we saw what is a sonicator. If you're sonicating your system or you're sonicating the cell, at that time, because of the high frequency of sound, heat is generated. Okay, and your suspension, your whatever biomolecule is now, it should have been at a certain temperature for stability, but now the temperature has increased because of sonication. So in such a case, the buffer should have the minimal effect. Okay, so we can see that temperature affects the buffers. Usually whenever you make buffers in the laboratories, they are stored in the refrigerators so that the temperature does not affect them, and also there should be no bacterial contamination in your buffer. So these are the two purposes why most of the time the buffers are kept in cold conditions. Okay, so generally there are two assumptions while you are using a buffer. There are two assumptions, is that the activity coefficients of the buffer ions is approximately equal to 1 over the useful range of buffer concentration, or the value of the Ka. What is this Ka? We just saw what is Ka. It's a constant, it's an equilibria constant over the working range of temperature. So these are the two assumptions which are done when you're working with buffer. But however, in the real practice, pH will change with change in temperature. Ideally, pH should not change. Okay, the assumption is that the pH is not going to change, but pH will change. Very critical in biological systems where a precise hydrogen ion concentration is required for reaction systems to operate with maximum efficiency. When we talk about pH, if you remember the Arrhenius theory, which you studied somewhere around in 10th standard, what is pH? Is that basically the concentration of H plus ions. Okay, so always I write it in square bracket like this. Okay, so pH change means what? The concentration or the number of H plus ion will change in the system. Okay, so if temperature rises of your system in which the analyte is there, then the hydrogen ion concentration also will change, and that will lead to the change in pH. So every biological system has a very critical requirement for the H plus ion concentration. If this H plus ion concentration will increase because of the change in temperature, or it will decrease because of the change in temperature, then the pH will also change. If the pH changes, then the cell will find it difficult to perform that particular reaction. Okay, so that's why temperature is very important. But generally the two assumptions are that buffer is so robust that it will, at under any conditions, it will not change the pH. Under all the circumstances, it will resist the change in pH. But ideally, it is not happening in real practice. There is it is observed that with change in temperature, pH also changes. Now, effects of buffers on certain factors. So there are certain factors. Okay. Now, other than pH, it is of utmost importance that the researchers establish the criteria and determine the suitability of a particular buffer system. So when you're working with a particular buffer system, you must also see that not all buffers are going to have the same, will behave the same in different conditions. Okay, so a phosphate buffer is maybe at a certain in certain condition the phosphate buffer performs fantastic, whereas in other conditions you will see that phosphate buffer doesn't perform very good. So it is up to the researcher that the researcher will have to establish the criteria and determine the suitability of a particular buffer system. So if the researcher is working with, say, a cell isolation or mitochondria isolation, something like that, then the buffer system used will be different, and the researcher must be aware of it. He or she will be establishing a criteria for determining the suitability. Now, some weak acids and bases, because I said no, that you will have a weak acid and a conjugate base when you're making a buffer system. So here, if there are some weak acids and bases which are forming your buffer, they may interfere in the reaction system, okay, because they are going to be a part, that is, buffer will not leave the biomolecule at any step. You will have to suspend the biomolecule in the buffer, because for the you want the stability of your biomolecule, and your biomolecule is very happy in that particular buffer, in that very particular pH. Okay, so but and you can't avoid the fact that they may interfere with the reaction system. So for example, if you're using a citrate and phosphate buffer, they're not recommended for systems which are highly calcium dependent. Okay, citric acid and its salts are known to be powerful calcium chelators. So now when you add a citric buffer or citrate buffer or a citrate phosphate buffer to a system in which calcium is going to be there, okay, then what will happen? It's a property of citric acid that it will go and bind with calcium. So if it goes and binds with calcium, it means that the calcium which was supposed to be available for something other, some other reaction, is not now available because it has been already in a bound form with citric acid. So in this such a case, you cannot proceed. Okay, so that's why if you're working with calcium dependent substance reactions or mechanisms in which calcium is important, freely available calcium should be there for certain reactions to happen, and if you are doing that particular work in the presence of citrate and phosphate buffer, you will see that this calcium will not be available for the other purposes, because calcium goes citric acid binds with calcium, it has an affinity to this calcium metal, so it goes and bind, and now the calcium is not available. Phosphates react with calcium, so phosphates also, citrates also. So now you know that if you're working on a assay system, for example, if you have isolated an enzyme and that enzyme gives you best activity in presence of calcium, okay, so so calcium is one of the important cofactors also for the enzymes. So calcium, there are a lot of enzymes which work very good in presence of calcium, so they are calcium dependent for enzymes for their optimum function. Okay, so calcium dependent enzymes will need calcium, but if you're using a citrate buffer or a phosphate buffer, then what will happen? That calcium will not be available for the enzyme, but it will be bound to this calcium, citric acid or to phosphate. So that's why you must avoid citrate and phosphate buffer if you're working on a calcium dependent system. Phosphates react with calcium and they produce insoluble calcium phosphate, and it precipitates out of the system. One of the big no-nos in an enzymatic assay is that your buffer on reacting shouldn't your buffer is inert, it has to be inert. But if in an assay procedure, if you see precipitation happening, that precipitation is unwanted, okay, that's undesirable. I will say the precipitation is undesirable, so you must avoid that, and you must know what is the purpose that you're doing, what is the purpose of this particular experiment, what are the components of this particular assay. If your assay is going to have calcium, then avoid citrate and phosphate buffer, use something else which will give you the same pH. Okay, there's a lot of variation, combinations of weak acids and certain base conjugates, and you can get a variety of pHs. So it's not that only citrate and phosphate buffers are there for this particular pH. You can use different buffers also based on what is the ingredient in your reaction. Phosphate ions in buffers can inhibit the activity of some enzymes such as carboxypeptidase, fumarase, carboxylase, and phosphoglucomutase. Okay, so phosphate buffers are usually preferred, but if you're using with, say, so if you're working with amylase, which is comparatively robust, if you're using lipase, protease, okay, some proteases, you can use phosphate buffer, but not, this cannot be applied to all of them. Okay, if phosphate buffer is working with amylase, lipase, and protease, it doesn't mean it is going to work with a huge range of enzymes. There are certain enzymes where you will see that phosphate buffers will inhibit their activity, which is carboxypeptidase and all that. Why? Because in an assay, your main intention is to find out the activity. Your main intention is to find out an optimum condition for that enzyme to give maximum activity. When I say activity, what do you mean? I told you last time that every enzyme has a substrate. So E is the enzyme substrate. It forms enzyme substrate complex and it will give you a product. Okay. So this complex keeps on. So again, there is also, there you will see association first. Okay, it's not dissociation. You will see association and you will see, then it can revert and it can dissociate into enzyme and substrate also. So this is the reaction. Okay, now you will have to find out a optimum condition where you get maximum product. Okay, if you have isolated an enzyme, you are going to first find out an optimum substrate for your enzyme. You're going to figure out an optimum reaction condition. What is the pH at which my enzyme binds perfectly well with the substrate? What is the pH at which the reaction proceeds forward and gives me a product? What is the pH at which the enzyme remains stable? Okay, so all of these conditions you are studying. So your main intention when you are doing an enzymatic assay is that, first of all, you are finding out the concentration. Second of all, you are creating optimum conditions for your enzyme to perform the best, and form a good number, a good concentration of the product also. That is the main intention. But if your buffer system is inhibiting the enzymatic activity, it means that then the enzyme substrate complex formation will be less. And if the enzyme substrate formation is less, then the product form will also be less. Okay, so more the ES complexes form, more will be the product also formation. So you must encourage your enzyme to make more product rather than inhibiting, rather than discouraging. And how will you discourage? By using wrong buffers for the enzyme. So you must, the researcher has to establish the criteria and determine a optimum buffer system for his or her biomolecule of interest, or the system on which he or she is working. Okay, now factors affecting the analyte structures and stability. We saw now which were the factors for that could affect the buffer. The pH is one factor, temperature is one factor. Okay, but when it comes to the analyte, knowing what your analyte is, knowing what the effect of temperature and pH on your analyte will also help you to decide what kind of buffer you are going to use. If both your buffers and your analyte is also sensitive to a certain thing, then you should avoid it. So because now we are talking about biochemical reactions, we are talking about cellular components. So buffer system is something that you cannot avoid. So buffer system has to be optimum. Along with the buffer system, now you should also know that what are the factors, what are the different parameters that are going to affect the analytes that you're working on. It could be a mitochondria, it could be a hormone, it could be a drug intermediate, it could be a metabolite, and it could be a protein or enzyme. It could be anything, okay, which you are extracting from the source, which you're extracting from a plant or an animal cell or a bacterial cell. Okay, so the importance of the structure of a particular analyte is important, because the function is always associated with the structure. Okay, the function of a particular analyte is always associated with the structure. It's associated with structure. The shape of, now in this case, let us consider protein. So the shape of a protein, or the shape of that enzyme, all enzymes are protein, okay, but all proteins are not enzyme. So the shape of this particular, say, protein is critical to its function, because it determines whether the protein can interact with other molecules. So last time, if you remember, I had mentioned about something known as a catalytic site. So every protein will have a catalytic site. This is the site where this is like an interaction point of the enzyme protein with substrate. Okay, so in the digestive system, we in your school that you have studied about salivary amylase. So the salivary amylase is present in the mouth. Okay, and it interacts with the starch in the food. So amylase is the enzyme and starch is the substrate. Okay, yes. So if now this amylase is in a proper shape, then the catalytic site will also be in a proper position, in a proper shape, right? So the substrate molecule has a they're complementary. So the substrate molecule is finding that catalytic site and coming and binding there. It's just like a lock and key kind of a procedure. So every enzyme has a specific substrate. So starch will find that compatibility with the catalytic site in the amylase and it will fit there, and the enzyme substrate complex is formed, and then the starch is hydrolyzed by amylase and a product is formed. That is the first step that happens in the mouth. Okay, now if imagine if that amylase shape or structure is distorted, is damaged, is distorted or it is damaged, then what will happen? The structure is damaged. It means the catalytic site also will have some damage. And if the catalytic site damages, the shape of the catalytic site will change. The amino acids which are there in the catalytic site will rather be removed. Okay, so it means now that the starch will not find compatibility with that particular catalytic site. Maybe it will go and fit inside, but since the amino acids which were required there are not there anymore, then there will be no further reaction that is happening. Okay, so that's why the shape of a protein is very critical. If the shape of the protein changes, the function of the protein also will change, because the protein interaction with other molecules is going to change. It is not that the protein is just happily sitting in the cell. It has nothing to do with others. No, there is interaction happening with between all the biomolecules which are there. So the protein will not be able to interact if the structure changes. So stability is very important. It is very important to know how stable the protein is. To what changes in parameters will the stability change of the protein. Now, will the protein retain functions and structures at a high or low pH and high or low temperature respectively? That is something for the researcher to find out. That if the researcher is going to change the pH of the system, then will the protein be able to bear that system change, sustain in that particular system? Will it be still doing the reactions that it is supposed to do the reaction? Will the protein remain the same? Will it maintain its structure at a change in temperature? That is something that every researcher has to find out. Okay, so the most important thing why we are studying about pH and temperature and the effect of it on an analyte is because when we talk about enzymes, when we talk about proteins, we know that they are very important in a cell because they are biocatalysts. We all know what is catalyst. Catalyst enhances the rate of reaction. So there are chemical catalysts, there are biochemical biocatalysts. So enzymes are known as biocatalysts. Okay, this is very important property of the enzymes, and that's why they play a very significant role in a proper functioning of the cell, because whatever reactions are happening in the cell, they are all catalyzed by these enzymes. Ability to increase the rate of reaction in every living organism, whatever reactions are happening, if you want to enhance the rate of reaction, then you need these biocatalysts. Okay, they must come and enhance the rate. So the enzyme, the substrate, sorry, not the enzyme and substrate, but if A plus B is the reactant and it is giving you C as a product. So if this is going to take 100 hours, but when A plus B reacts in presence of certain enzyme, it might take only 1 hour and you still get C. So which is more economic, feasible, efficient? Of course, this So who intervened? Who came and enhanced the rate of reaction? It is the enzyme who came and enhanced the rate of reaction. This enzyme, after it does the job, leaves the reaction and goes and participates in other reaction. So this enzyme is reusable. It is inert. It will not interact or it will not interfere in anything, but it will only increase the rate of reaction. You'll get more product. So if in 100 hours you were getting only 1 mole of C, so now you're getting in 1 hour you're getting 1 mole. So in 100 hours, because of this enzyme, you'll get 100 moles of C, and this is going to be so productive. Okay, so industrially important enzymes and all that, they would want that their reaction rate is increased and they get more product. Okay, so enzymes are biocatalysts, very very important. Now most of the enzymes are proteins. Okay, their activity is affected when their structure changes. When the structure changes, it means that the amino acids also are going to get modified, because a protein is made up of amino acids, and if the amino acids change, the protein will also change. The shape will also change. The interaction capacity will also change. Okay, so that's why the shape of that particular protein or the structure of that particular protein is extremely important. But what are the parameters which are going to change the or disrupt the structure of the protein? It is change in temperature, it is change in pH, and it is the reactant or the substrate concentration that is going to affect the activity of the protein. It is also going to affect the structure of the protein. Okay, so enzyme activity can be measured by monitoring either the rate at which a substrate disappears or the rate at which the product forms. Okay, so if you are doing this reaction, again E plus S, reversible, ES, and it will give you, this is a reversible step activity protein. Okay, now there are two ways to find out whether the enzyme is active or not. How will you find out? So if I have 10 substrate molecules when I started the reaction, okay, after I finished the reaction, there is only 2 substrate molecules remaining, and say suppose there are, and when I started the reaction there was no product, right? When you are at the initial step of the reaction, you have just added the enzyme and the substrate, there is no product. But after some time you see that substrate concentration has reduced to 2 and the product concentration is now say 8. Okay, so it means that the substrate concentration has decreased and the product concentration has increased. So your enzyme is active. So your activity of the enzyme can be measured by monitoring either the rate at which a substrate disappears. So yes, in how much time. Now rate means in how much time did the substrate concentration come down from 10 to 2, whether it took 8 hours, 8 minutes, or 8 seconds. In how much time did this substrate disappear from 10 to 2? In how much time did this product form from 0 to 8? So that will help you to know whether your enzyme is active or not. Okay. So temperature is extremely important. It is very, in practical applications also you will see how to relate that effect of temperature on certain things. The most of the chemical reactions you talk, when you increase the temperature, when there is a rise in temperature of 10-degree rise, okay, so if you're doing a certain reaction say at 40 degree, and if you rise the temperature to 50, you will see that the rate of reaction is doubled. The time taken for the reaction will minimize. It will be faster when you increase the temperature. Now, to some extent, even enzymatic reactions also will show this behavior. So, as soon as you increase the temperature, you will see that the enzymatic reactions have also doubled the efficiency. The rate is double, but at a certain point, it starts decreasing. Okay, it's not that if you're doing an enzymatic reaction at 40, and you raise it at 41, then 42, then 43, then 44, then 45, you will see till 45 there is an increase, but then it will start dropping from 46, 47, 48, like that. So enzymatic reactions do not constantly show a higher rate of increase in reaction with increase in temperature. At after some point the rate decreases. So that is because now at higher temperature the protein is not able to maintain its structure. The protein has denatured after a certain rise in temperature, and that's why now, because of the denatured structure, there is a disruption of the active site. For many proteins, denaturation occurs between 45 and 55 degrees. Okay, so if you look at this graph, you started, this is the place you started, 0 degree. Now you started increasing the temperature of your enzyme, okay, of the system in which your enzyme is. And because of the rise in temperature, you will see that the more and more substrate and enzyme complexes are forming, and at there is a point here you will get the maximum activity. Activity means what? The rate at which the substrate disappears and the product appears, the rate at which. Okay, now you have found that this is the peak. Now what is that temperature? If you look at it, if you just, so maybe somewhere around 35. So 35 is the optimum temperature. Now if you still keep on increasing the temperature of your system, okay, what is this system? This system is enzyme plus substrate plus buffer. Okay, it's a buffer system of obviously. And then you are encountering more and more product that is being formed till 35. After that you will see that there's a decline in the rate of reaction. No more products, or very less products, very few product molecules will be formed, and the protein at one point will become so inactive because it has lost its complete structure. Now nothing will be happening, no reaction will be happening. So most of the biochemical reactions are carried out at lower temperatures because of the issue of the stability of the enzymes. At higher temperatures, enzymes will denature after a few minutes. That's why in the practical applications also you see that we usually sterilize objects that we are using. Even at home also we boil the water, we for sterilizing, or even in the medical practices also you will see that happening in the laboratories also, while you're using an autoclave where you are actually heating, moist steam has been given to the objects to sterilize them, because at a higher temperature if there are certain bacteria, okay, these bacteria are going to have enzymes within them. So at a higher temperature the enzyme within the bacteria will be denatured, and because of the denaturation of the enzyme, now there is no further activity that is going to happen. The cell will be dead. Okay, so you are killing the enzymes, you're denaturing the enzymes at a higher temperature. We sterilize, that's why we use higher temperatures to sterilize objects. Now lower temperatures, for example the home cooked food that is kept in the refrigerators, you keep them at lower temperature, you freeze them, because you're slowing the activity of the enzyme, because at a certain temperature the enzyme activity is at its peak. So look at this graph. At 35 it was maximum, but how was the activity at 4 degrees? So 4 degrees, say maybe somewhere here, this one, this part, this is the 4 degree, which is very less. So what are you doing? By reducing the temperature you are reducing the activity of the enzyme also, so the food will stay eatable for a longer time. Okay, but if you keep the same food outside and at room temperature, the temperature is say between 30 to 35, the enzyme activity is more, so enzymes, the bacteria will flourish at that temperature because the enzymes are very active. The bacteria is going to chew up all the food, and it will be it cannot be eaten by us then. When animals, a lot of animals go into hibernation also in the winter, bear being one of them. So why do they go to hibernation? Because they want to avoid the cold, severe cold outside. Because of the severe cold, the body temperature will also drop. If the body temperature drops, the metabolic processes which involve the enzymes participation will also drop down. Okay, so it can be, then what will happen is, see, when the temperature outside is increased, the temperature inside should also increase. The body will have to work faster and faster to create more heat. Okay, so to keep the body warm. But when they hibernate, then they don't have to generate so much heat, because of obviously they are not exposed to cold, they are finding a warmer place and they are hibernating in that warmer place. So then they will not have to generate more heat, which means that they can store the fat reserves. They will have to use lesser fat reserves to keep themselves warm. But if they go out in the cold, more and more fat reserves will be used up to keep their body warm. Okay, so these are some of the applications which we see of temperature in the practical life, day-to-day life. Effect of pH is another important. So every enzyme has an optimum pH, optimum temperature. What is the optimum pH, or what is an optimum temperature, or what is basically an optimum condition? So every enzyme will have an optimum condition where it behaves the best, gives the maximum activity at that pH. Okay, so there is an optimum temperature also, where it at that particular temperature the enzyme is going to behave the best. At that particular pH the enzyme is going to behave the best. So it is known as optimum condition. Okay, now you have studied that trypsin and pepsin. These are the enzymes, very important enzymes in our digestive system. They are important because they break down the protein chains. And this protein is a complex molecule. It breaks it down into simpler or smaller fragments or pieces, or into smaller peptide chains. Okay. And these peptide chains then are further broken down into individual amino acids. Now, pepsin is in the stomach, the pH is acidic, right? And then trypsin is alkaline. See, both these enzymes work on protein. So both of them have protein as a substrate, but yet both of them have different pHs. So it means that if I change the pH of pepsin, so if pepsin optimum is 1.5 to 2 and trypsin say somewhere between 7.5 to 8, this is the optimum, this is the best pH at which it will perform the best. Okay, but if I change, so if I make pepsin 8, if I subject pepsin to pH 8, and if I subject trypsin to pH 2, they will not function. So it proves to us that pHs are the most important factor along with temperature. Okay, so there's enzymes are sensitive to pH. You look at this particular graph. So if suppose this is 0, if this is 1, 2, 3, 4, 5, 6, 7, 8, you will see that this is the peak here. This is known as a bell curve. Okay, you'll see the peak here, which tells you that at pH 5 the enzyme was the most active. So this becomes the optimum pH. Okay, so they are sensitive to the change in pH. The change in pH of the surrounding will also change the shape of the active site. That is again very, very important. The active site uh, shape changes, and hence now it will affect the interaction of that particular enzyme with a substrate. Many amino acids in an enzyme molecule will carry a charge. We saw that amino acids have charge, and these are responsible for the interaction within them, and that is why a particular shape comes to the protein. The folding happens of the protein, okay, because there are these amino acid interactions that are happening. Now within the enzyme molecule there will be positively, negatively charged amino acids. They will attract each other, and hence they will form a binding, and this kind of interaction between the positive and the negative charges will contribute to the folding of the enzyme molecule, and that folding is going to decide the shape and the structure of the active site also. Okay, every enzyme is having a active site. Now change in the pH, how will it affect? The change in the pH will affect the charges on the amino acid molecules. Now amino acids which were attracted towards each other, because of the change in pH, will no longer have any attraction. If there is no attraction, if there is no interaction, then the structure also will loosen up. Okay, so the shape will change, the active site conformation will change. Now if the shape is changing, the catalytic site is changing, or the active site is changing, it means that the protein is not the enzyme will not be able to interact with the substrate. So it means that there will be no reaction. So this pH is a denaturant, or extreme pH will denature the enzyme. So the changes are usually um, the changes happen when you change the pH. You will see that the enzyme is affected. The activity of the enzyme reduces. Okay, but sometimes it might lead to a permanent change, and sometimes you'll also observe that when you bring the pH back to the optimum, the enzyme is functioning very well. Okay, that is, the enzyme is tolerant to change in pH. Same will be observed in change in temperature. If you change the temperature, if you change the optimum temperature, you will see the enzyme is not performing well. But when you bring back the optimum temperature, the enzyme starts performing well. It means that even if the temperature had risen or it had lowered, it could not change the active site. It means that the protein or the enzyme is tolerant to a slight change in pH and temperature, but quite a few of them will be sensitive also. Okay, so with this we stop today, and we will look at the other sub parts in the other class. Okay,